All right, so I'm Nick Sather. I'm the co-founder and CEO at AmphixBio. And first off, I'd like to just thank the organizers of this conference for the invitation to present here today. I'm excited to introduce you all to a new therapeutic modality that we're developing to treat spinal cord injury. So I started AmphixBio with my PhD advisor, Sam Stoop at Northwestern University, with the goal of creating a new class of regenerative therapies to restore lost function, whether that's due to injury, disease, or aging. And we started this company focused on musculoskeletal regeneration at first, but we've since moved towards neurological regeneration. And so all of the diseases that you see here are areas that we're targeting with this new therapeutic modality. And we have a specific interest in spinal cord injury, which is what we're going to talk about today. So typically when you think about regenerative medicine, you think about cell or gene therapies, and that's for good reason. They're highly potent. But if you look in the clinic, they've been relatively limited, and that's mostly due to manufacturing scalability and some reproducibility issues as well, going from small animals to larger animals. And on the flip side of this, you have traditional biomaterials or small molecule, large molecule therapeutics. These are much more scalable, but often lack the true potency needed for regeneration. So where you want to be in this 2x2 is in that upper right corner, and that's where we focus. So our approach is to use patients' own cells to trigger regeneration of new tissue. Now, when you think about cells, they're not just in our bodies by themselves. They're surrounded by what's called the extracellular matrix, or ECM. And this is not just a physical scaffold, but it's also a biochemical support. And so when we're developing our therapies, we're thinking about everything around the cells to really, truly drive regeneration. So our technology is based on these programmable peptide molecules that are encoded with both structural and biological information. So this blue segment that you see here is a series of amino acids that are designed to encourage self-assembly of these molecules into these long fibrous nanostructures that look very similar to the natural ECM that we looked at in the last slide. But on the surface of these structures, we have this green segment that is a biological signal that directly interacts with cell receptors. So these are what we've coined supramolecular therapeutic peptides, or STPs. And this technology has leveraged the last 25 years of development in the Stoop lab at Northwestern. There's over 30 issued patents, hundreds of papers published in some of the highest impact journals, and we've made thousands of these molecules to date. So how these work, and this is really the key takeaway here, is that this is a two-in-one therapeutic. It is both a receptor activating drug and a tissue scaffold for regeneration in the same therapeutic. So on the signaling side, what you're looking at in this cartoon here is the cell membrane in light blue. You have transmembrane receptors in dark blue and gray. And then you have a green signal floating around the extracellular space. This is how traditional drugs are designed, right? So you're just hoping that that green signal comes and interacts with the right receptor to turn on that signaling pathway. But in an STP, now you have that green signal localized on those fibrous nanostructures. They're presented at very high density and they're near the cell surface. So this really allows for receptor activation and clustering and much more potent regenerative pathway activation. On the scaffold side, when we're looking on the right-hand side here, and the very far right is an electron micrograph of natural extracellular matrix in the spinal cord. So if you took out all the cells, what would be left behind is this fibrous tissue. And that's what you see here. And on the left, you see a blue neuron growing on an STP scaffold. And what you'll appreciate here is that the three-dimensional architecture is very similar. These are designed to have very similar mechanics to natural tissue. And they form a hydrogel upon administration for cell infiltration and ultimately regrowth. Now, this is truly a platform technology, right? So what we're focusing on to start, our lead vertical, is using these peptide scaffolds as regenerative drugs. And so, we have positive efficacy data in these various indications listed on the bottom here from SCI, stroke, ALS, and also other musculoskeletal indications as well. But I think this is very important for this audience today is that we have also secondary verticals using this technology in combination with other modalities. So we also have preclinical data using these in combination with growth factor therapies, with cell therapies, and also with peptide drugs. And I think this technology in particular lends itself very well towards these combinatorial approaches. And there, I think, could be very synergistic applications here as well moving forward. Okay, so our lead asset is called AmphixBio, and we think that this will really be a paradigm shift for treating neurological disease. We do that through multiple mechanisms of action. So we'll walk through these on the next few slides, but there are several things going on here. There are multiple molecular targets in this therapeutic. So this is both an agonist for FGFR1 and integrin beta-1. The signals are presented dynamically on the nanostructures to cell receptors, and they form this physical scaffold that matches the porosity and mechanics of native tissue. As far as the mode of action, this therapeutic works on multiple cell types. So it's not just neurons. There are receptors for these targets both on glial cells and endothelial cells as well. And we've seen neuroprotective effects, neuroremodeling, and also neuroregenerative effects with these therapeutics. And I'll walk through the data in the next several slides here. So our lead indication for this asset is acute spinal cord injury, but we've also used it in follow-on indications in chronic SCI as well as ischemic stroke and ALS. And we've seen some positive efficacy in these as well, which gives us some confidence that what we're seeing is a real effect in terms of the neural potency. Okay, so from the target's perspective, SCI is a very complex injury, and I think this audience is very well aware of that, right? Glial scarring is a major issue that inhibits regeneration after SCI, and on poor vascularization, the loss of trophic support obviously leads to cell death. So our approach to combat these issues is to have a multiplex targeted approach, right? So our first signal here is a laminin mimetic peptide called the IKBAV, and this is an integrin beta-1 agonist. It's designed to limit the glial scarring and also encourage the axonal outgrowth after injury. Second signal is an FGF2 mimetic peptide. This is an FGFR1 agonist, and this one is designed to promote blood vessel formation as well as the survival of neurons. So these two signals have been combined together into one STP here called AmphixBio, and the ratio of the two was heavily optimized in a science paper in 2021, and that's the basis for this technology. Okay, so this is the next part is around the dynamics, and this is very unique to this therapeutic modality, right? So if you were to look at an STP nanostructure here on the left, and you cut that in cross section, what you'd see are these molecules arranged in this manner here. Now, what's very unique here is that because these molecules are not covalently bound to each other, they can actually move along the surface of the structure and also extend in and out of the nanostructure surface. And that's because they are supramolecular interactions, not covalently bound. And why that's really important is on the receptor signaling side. So in comparison to covalent molecules, right, your receptors or your signals are locked in space. Here, they move along the surface and they can really highly influence clustering of these receptors together, which is really important specifically for these two targets that we're going after. And that is the key differentiator between this technology and also why it works a lot better than just these molecules alone. Okay, last piece is around the administration. So AmphixBio is a viscous liquid in the vial, right? And so when this is administered, it's a 99% water solution, but when it touches tissue, then it forms this hydrogel. And so why this is important is that it can be delivered through very small bore needles, mini bore tubing, and we've done that. You can see in our rodent models here, but this is also very compatible with what's been done in clinical trials with cell therapies in these intraparenchymal injections. The other piece here is that compared to standard scaffolds where they have a physical geometry of volume that is defined before implantation. Here you can minimally invasively apply the therapy, and it will space fill in the lesion to form that hydrogel without damaging and resecting the damaged tissue around it. All right, so now let's get into the data. This is what we're very excited about. So we validated the efficacy of AmphixBio in multiple rodent models of acute SCI. So on the left here, this is a mouse model that was done in the Stoop lab. And what you'll see here in this first video is a completely paralyzed mouse. It's dragging its hind limbs, no weight bearing whatsoever. And then with a single injection of this therapeutic 24 hours after injury, this mouse is now walking again, right? So there's plantar stepping, there's weight bearing. So we were very excited about this. You can see that in the BMS scoring for locomotion on the left here, where we see within 4 weeks, significant improvement in locomotion and also sustained over 3 months. This is also borne out in the gait analysis here in these ink print images. Now, on the right-hand side of this slide, we also reproduced this in a rat model at a CRO in France. And so they saw similar improvements in BBB score, again, with a single injection 24 hours after injury compared to injured animals. We also did not see any neurological deficit in healthy animals when you're applying the therapy via intraparenchymal injection. And they also did a battery of safety assessments on these uninjured animals. So we did not see any neuropathic pain or immunotoxicity or spasticity associated with the injection, which suggests that it has a clean safety profile. All right, so this was backed up, though, by strong histological evidence of these mechanisms of action. So there's 5 key effects that AmphixBio has in acute SCI in tissue. So we'll walk through each one of these individually. So the first is in corticospinal tract regrowth, and so you can see that in the top right over here. So compared to sham, what we have is BDA-labeled axons from the brain through the injury and into the distal border of the lesion. So we see that clearly in these red tracks across through the lesion center and on the other side of the lesion. We also saw a significant reduction in glial scarring at the border of the lesion, which you can see on the bottom here. And again, you can see these BDA traces going across the lesion site through to the distal border. Okay. The other pieces here are when we zoom in and we look at those axons that are regenerated compared to a control therapy that has the slow motion of the molecules. When we use the Amphix 200 with the fast motion molecules, now we have a significant increase in the amount of myelin basic protein that's surrounding the axons. We also looked at dye-eye labeled blood vessels that were formed within the lesion. And so you can see here compared to the sham controls or sham injured animals, there's a significant increase in functional blood vessels through the cord in these transverse sections, similar to uninjured animals. And finally, when we looked at neural cell survival, both just total neurons and new end staining, as well as chat motor neurons in the ventral horn, we see a significant increase compared to the sham saline injected animals when we had this Amphix 200 injection too. So we're very excited about the results in acute spinal cord injury, and we're pushing that forward as fast as we can to get that in the clinic. But we've also tested this in chronic SCI. And so this is active area of research in the Stoop Lab at Northwestern. So I'm just going to show a quick preview of how that data is looking so far. This is an early pilot study, but we're excited about this too. So this was a severe chronic SCI model. This is a T10 contusion. We wait 8 weeks after injury before applying Amphix 200. And then what we see is within 4 weeks, we do see an improvement in BBB score. Again, the baseline BBB for these animals is about 2 to 3, so they're heavily impaired. And then within 4 weeks, we see this improvement in locomotion. But we were also very excited when we started looking at the histology here. So compared to the saline controls, the ones that these are chronic injured animals that get the saline injection 8 weeks after injury, we still see this large hole, the cavity in the middle of the cord. But when you compare that again to a single injection of Amphix 200 into the cavity, we see that that is now filled with new tissue, and it's stained with TUJ1 neurites, as well as serotonergic 5-HT neurons in the lesion cavity. So again, this is early days, a lot more work to be done here to figure out the mechanisms of action. There's a ton of work being done in the Stoop Lab as far as improving this therapy, adding new signals to make it more specific for chronic SCI. But I think even with the original therapy, we're very excited about the potential here. So as far as the pipeline goes, I talked a lot about acute SCI today, a little bit about chronic SCI, but we also have, some very promising data in other neurological indications from stroke, ALS, Parkinson's, and neuropathies. But our goal is to get this acute asset into the clinic in a phase 1 trial by the end of 2026. We also have a musculoskeletal regeneration program. Our Amphix 100 is another asset that we're developing for degenerative disc disease. And that one recently got an FDA breakthrough device designation, and that we're looking for partners to develop that and move that into the clinic as well. All right. And I think, we have the team to do this. So we have a very specialized, deep domain expertise on the science and tech side. Sam Stoop is really the leader in supramolecular chemistry and its application to therapeutics. We're supported by some of the leading clinicians in the space, including Jim Guest, who's been extremely valuable in our design for the phase 1 trial. And then we have a great group of industry veterans, have been in the space for many years who are advising us on the business side as well. So I'm going to close here. Again, I want to thank the organizers for the invitation here and just reiterate our dedication to trying to make a therapeutic for spinal cord injury. We know it's going to be a long road, and it definitely takes a village. So please do connect via our website or via LinkedIn. Would love to connect, and I very much look forward to the rest of the conference. Thank you.